our multi-choice for our booklet here. Question 1. An unfavorable mutation in tumor suppressor genes is unlikely to do what? Now, let's just really be careful of the wording here. We're looking for unlikely, so not likely. Tumor suppressor genes essentially code for a protein, a specific protein with some specific functions as well. For example, initiating apoptosis and decreasing the rate of cellular division, which means that if a mutation does happen in it, the function of this protein won't occur. So in this case, it is likely that a mutation will prevent cells from responding to apoptotic signals, and it is likely that it will increase the rate of cellular division, and hence it's also likely that it will result in a breakdown of the regulation of the cell cycle, leading to cancer, as we were looking at before. So in this case, it's unlikely that our tumor suppressor genes will transform into oncogenes. A smooth muscle cell has reached the G1 checkpoint of the cell cycle. So G1 checkpoint is a really important bit of information. So in my head, I'm already thinking, let's just try to picture a diagram of the cell cycle. Well, we know that there are 4 main sections. We've got the G1, got the synthesis phase, the G2, and our mitosis phase. And our cell cycle goes this way, all the way around. And our G1 checkpoint would obviously be in the G1 phase. And if we want to think about what's after the G1 phase, it's the synthesis phase. So this is where DNA replication is occurring. So trying to keep that in mind, that really helps us to think about, what's the purpose of the G1 checkpoint? It's obviously to assess the DNA before it's being replicated. We're just going to look at identifying each of these different cell organelles. So let's just run through the list. So first of all, we've got a ribosome. So these are all the small little dots, like right here. They can be either found in the cytosol, in the fluid, or attached to these tubular networks known as the endoplasmic reticulum. So this is our ribosome. We've got our nucleus, which is the storage of the DNA, the site of the storage of the DNA, and it's this central sort of really important aspect of the cell here. And let's not confuse that with the nucleolus. So it's this section, which is the nucleolus. And that is where our ribosomal RNA is made. So over here,